Yeah.
A very good evening, everyone, and welcome back to again our fifth lecture of this monthly research article spotlights. So today's lecture is by Mr. Soham Chowdhury. He will present his work on the composition dependent formation and dissociation of structure 1 and structure 2, Clathrate hydrates of trimethyl oxide in ultra high vacuum. So Soham did his Mola, uh, bachelor's from Maulana Azad College from University of Calcutta and master's from IIT Madras. After that, he joined Professor T. Pradeep Lab in IIT Madras. So welcome, Soham, and the floor is yours. So please restrict your presentation to 25 minutes so that we have the plenty of time for the discussion. So
Thank
floor
you,
is
Deepak, for this nice introduction. So let me share my screen. I hope it's full screen now.
yeah, it's full screen.
So thank you all for joining today. So welcome to this monthly spotlight lecture. So today I'll be presenting a paper recently published from ICE Lab in JPCC Journal and in 5th May of 2025. The paper is titled as Composition Dependent Formation and Dissociation of Structure 1 and Structure 2 Clathrate Hydrates of Trimethylene Oxide in ultra vacuum. This paper is co-authored by Dr. Bijesh. He is now a postdoctoral fellow in our lab. Dr. Gaurav Bhaiya is now in NUS, Singapore, working with Dr. Provilinga. And Anupriya Ji was a ex alumni of our lab, postdoctoral fellow, and he helped us in the computational study. So let's get started with the.
So Gaurav has also joined, so let us welcome him. Yeah.
Yeah, I have sent invitation to every our ISAF alumni. So thanks for joining.
Very nice.
Good.
Glad.
So let me start with what is Clathrate hydrate. So Clathrate hydrate is a host-guest inclusion compound where water molecules comes together and creates a hydrogen bonded network. In this hydrogen bonded water network, small molecules can be trapped. So what water molecules here is the host and the trapped molecule is the guest. This kind of host cage structure forms a supramolecular cage that is of Clathrate structure. And as this Clathrate structure is made up of water, this structure is known as Clathrate hydrate. So this Clathrate hydrate can be of different sizes of cages like small cage or large cage. Small cages are mostly 512 cage, which is made up of 12 pentagonal faces, and large cage typically of 512-6-2 and 512-6-4 cages, where 512-6-2 means 12 pentagonal and 2 hexagonal cages, 512-6-4 similarly. So for structure 1 Clathrate hydrate, it is made up of 512 cage and 512-6-2 cage, and for structure 2, it is made up of 512 cage and 512-6-4 cage. Now let's look at this phase diagram, this temperature pressure plot, where you can see that to create Clathrate hydrate, we can create it at a relatively low temperature near about 0 degree centigrade, but it's require a high pressure. If we lower down the temperature, the requirement of pressure also goes down significantly. So theoretically, we can create Clathrate hydrate at very low temperature and also at very low pressure. So we can cut down the requirement from high pressure, relatively low temperature to low pressure, low temperature condition. Keeping this in mind, we have started our work. So, like as we can see on Earth during the winter season, in the Nordic countries and specifically in US, the pipelines get clogged with these big chunks of Clathrate hydrates. So it is usually seen on Earth, but in space or interstellar medium, how Clathrate hydrate can be formed, there is no direct experimental proof. So here comes our paper, this published in PNAS journal in 2019, where we have first established that Clathrate hydrate can be formed in interstellar medium condition. we have prepared methane hydrate and CO2 hydrate, and which is detected by the signature reflection absorption IR spectrum peak, which has shown that CO2 and methane molecule is going inside the Clathrate cage in the amorphous water ice. So following this study, so we have published quite a few papers on Clathrate hydrate. Now comes in our paper, which we have shown that TMO Clathrate hydrate formation. So why we have chosen this trimethylene oxide TMO molecule? For that, so ethers are common to form Clathrate hydrates. Like if you see the simplest ether, that is a dimethyl ether, the non-cyclic ether, that forms structure to Clathrate hydrate in the 512-6,4 case. Also, tetrahydrofuran, that's a cyclic 5-membered ether, that also creates structure to Clathrate hydrate. Now, if we see the ethylene oxide, that is a 3-member structure. So this ethylene oxide also forms structure 1 Clathrate hydrate in high pressure, also in high vacuum condition. Now, so we have talked about this 4-member ring and 3-member ring. So TMO falls just in between. It is a 4-membered cyclic ether. So whether TMO will form structure 1 cage or structure 2 Clathrate hydrate, or it will form both, that is not unanswered. That is not answered properly. And also, TMO is yet to be detected in space, but its structural isomer, that is propylene oxide, that has been detected in interstellar medium at 2016. So TMO would be an interesting molecule to study in for the purpose of interstellar chemistry and as well as Clathrate hydrate study. So there are a few reports on TMO Clathrate hydrate where they have created a TMO Clathrate hydrate at high pressure condition or at high vacuum condition by depositing guest molecule on the ice nanocrystal, and they have recorded the IR spectrum as well as the Raman spectrum. But there is no study of TMO Clathrate hydrate formation in UHV condition, as well as these are nanocrystals of ice. But what we have done here, that is starting from the amorphous water ices to the higher temperature and waiting there for longer time. I will describe that. So this is the background of the study. So the instrument that we have used, this is our SAT's brainchild. This was built in 2011. This is the ultra vacuum setup, where the inside the chamber the pressure is kept as 10 to the power minus 10 millibar. That is identical to interstellar medium condition. And also inside this chamber there is a ruthenium 001 hexagonal sample substrate that is attached to a liquid helium cryostat, and this stage can be cooled down up to 10 Kelvin. So this instrument comprised of many analytical techniques such as reflection, absorption, IR spectrometer, cesium ion gun to perform secondary ion mass spectrometry. It has temperature program resorption mass spectrometry, one deuterium continuum to perform vacuum ultraviolet irradiation. This is the MC detector to record the IR spectrum, and the main part of this is low energy ion scattering mass spectrometry to study the ion molecule reaction on the ice surface. So next comes to the experimental protocol what we have followed, and this also tells about the story in a nutshell. So we have deposited TMO and water molecule and different composition, that is 1:3 and 1:15 specifically at 10 Kelvin. After depositing the mixed ices, we have heated it up to 135 Kelvin at a 5 Kelvin per minute ramping rate and waited that for longer time. So we have seen that for these two different composition, in case for 1:3 ratio, structure 1 hydrate is forming, but for 1:15 ratio, structure 2 Clathrate hydrate is forming. And when we waited there for longer time, we have seen that for structure 1 Clathrate hydrate, after even after waiting for 17 hours, the Clathrate hydrate structures almost remains intact, and water also stays as amorphous ice. But for this structure 2 Clathrate hydrate, we have seen that the water amorphous water ice is being converted to hexagonal crystalline ices, but this is also the structure 2 Clathrate hydrate getting dissociated. So this is very contrasting result we have seen in this study. I will explain why and how this is happening. So let's start with the very basic study, that is the pure TMO IR study. We have taken the rare spectra pure TMO. We have seen that the amorphous TMO comes at 986 centimeter inverse at COC stretching region and 1026 centimeter inverse at ring puckering mode. So the main focus here in these two stretching mode, one is COC stretching mode, another one is ring puckering vibrational mode. So now comes here the study where we have taken TMO and water here together and a different ratio and heated it up to higher temperature. So figure A and B is talking about the TMO and water ratio at 1:3, where we have seen that at 0 hour there is at 135 Kelvin 0 hour, you have seen only one peak 1020 centimeter inverse. But after waiting for 2 hours, a new peak is appearing here, that is at 1035 centimeter inverse. This new peak corresponds to structure 1 Clathrate of TMO as reported in many other Raman study and IIR study. So we have further deconvoluted the spectrum and we have differentiated the two peaks, that is one is about this TMO Clathrate of structure 1 and another one is for crystalline TMO. Now this T and D talks about TMO and water at 1:15 ratio, where we have heated the ice mixture from 10 Kelvin to 120 Kelvin and waited there for longer time up to 18 hours. And we have seen an onset of a new peak at 986 centimeter inverse, which corresponds to structure 2 Clathrate of TMO. When we deconvoluted this spectrum, we have seen that there are three components present. One is structure 2 Clathrate of TMO, another one is crystalline TMO, and another small quantity is present, that is hydrogen bonded TMO. So TMO has an ethereal oxygen that can be coordinated with water hydrogen atom of water, and it would be hydrogen bonded TMO, and it's obviously redshifted because of the weakening of TMO bonds. So now another previous slides I have shown you the ring puckering mode of structure 1 and COC stretching of structure 2. But what about others? So this is the peak, this is the COC stretching mode of structure 1, where we have seen that very slight peak is coming from structure 1 TMO Clathrate hydrate. And also if you see for ring puckering mode of structure 2, the peak is also very less, the intensity is very less compared to the previous one. So here we have clearly seen that the formation of structure 1 TMO is more prominent in a ring puckering mode, but for structure 2 the formation of Clathrate is more prominent in this COC stretching mode. Why there is so different? Usually we are seeing that Clathrate for a particular gas molecule form shows their signature peak at a particular wave number region, but here it is changing the wave number region depending on the ratio. So we have taken help from computational study. So what we have seen that TMO cannot go in the 512 case because that is very small compared to the size of TMO, but it can occupy 512.62 case and 512.64 case. So as we are speaking, the structure 1 TMO Clathrate and structure 2 TMO Clathrate can be formed in the ice matrix. After that, we have looked for the binding energy of these structures and also dihedral angle. So what we have seen that structure 1 TMO is slightly more stable than structure 2 Clathrate hydrate. And when we have calculated the dihedral angle, we have seen that for structure 1 Clathrate of TMO, the TMO is not in planar condition. It is in distorted planar structure where the dihedral angle is around 9.282 degree. But for structure 2, TMO stays at almost planar structure. So here comes the answer, like as TMO is inherently a planar structure and also in structure 2, it is almost planar. So the COC stretching mode is mostly showing us the prominent peak for Clathrate hydrate. But when it goes for structure 1 Clathrate hydrate, because of this distorted structure, the ring puckering mode gets activated, because ring puckering is all about the distortion of the this cyclic ring. So as this ring puckering mode gets activated, that is prominently showing us the Clathrate formation rather than CO6 stretching. Now it comes at the dissociation of Clathrate. So we have created the structure 2 and structure 1 Clathrate and waited there for longer time period to see the dissociation of Clathrate and consequent effect of dissociation on the waterized matrix. So as you can see, I will first explain this figure B, where we have created structure 2 Clathrate, and you can see at 130 Kelvin at 0 minute, the water stays as a amorphous water ice. But when we have waited for up to 2 hours, the peak becomes gradually sharper and narrower, and which corresponds to the crystallization of water ice to amorphous water ice from amorphous water ice to hexagonal crystalline ice. So it is happening within 2 hours, and also you can see this Clathrate hydrate wave number region. So where we can see that this 986 peaks is continuously decreasing. So that is also indicating us that TMO Clathrate hydrate in structure 2 form getting dissociated, and TMO is getting out of the cage, and furthermore it is helping water ice to reorient itself and thereby converting from amorphous ice to crystalline ice. Now, when we look at this structure 1 cages, so here you can see we have waited at 135 Kelvin for 17 hours. So we have seen that there is a slight decrease of this 1035 peak, but even after 17 hours, a significant amount of TMO Clathrate present there after 17 hours. So that says that the dissociation of TMO Clathrate in structure 1 form is very slow and it is quite stable, as we have seen in the binding energy result. And now comes to the wave stretching mode, where we have seen that after 17 hours, the wave stretching mode is still very broad, and it stays as amorphous ice. It is not converting to hexagonal crystalline ice, and that is very surprising because if you look at this pure water rare spectrum, we have taken pure water 150 monolayer at 135 Kelvin and waited there for longer time period. So what we have seen that in the wave stretching mode, this first spectrum is amorphous at 0 hour, and within 12 hours, this water ice gets converted to crystalline hexagonal ice from amorphous ice structure. So water takes pure water takes only 12 hours at 135 Kelvin to being converted from amorphous hexagonal ice. But here, if we can see, up to 17 hours, the water still remains as amorphous. It's not getting converted to hexagonal. So what does that mean? It means that the structure 1 Clathrate of TMO and the TMO present in the ice mixture actually inhibiting the kinetics of this conversion and restricting water to go from amorphous ice to hexagonal ice. So water is staying at mostly in amorphous compact ice. It's not converting to hexagonal ice. So now comes at this kinetic study. So as we have seen that for structure 2 Clathrate, the Clathrate gets dissociated and converts the water ice to hexagonal amorphous from amorphous hexagonal ice. So we have studied this crystallization kinetics using Abrami equation. The first plot is about the linear plot of Abrami equation for this crystallization process, and second plot is about the Arrhenius plot, ln k versus 1 by t. From this kinetic study, we have calculated the activation energy that is required to convert amorphous ice to crystalline ice. So what we have done, we have performed the experiment in 1:30 ratio at different temperature, 128, 130, 133, and 135 Kelvin, and then collected all the data points and then plotted here. So the activation energy comes around 52.7 kilojoule per mole, and that is usual crystalline usual energy required to convert water ice from amorphous ice to crystalline ice. It falls in the range between 52 to 58 kilojoule per mole. So the key takeaways here same TMO molecule is occupying two different cages at same temperature pressure condition. So usually Clathrate hydrate is a phase of water where if we can play with temperature and pressure at different temperature pressure condition, we will get different Clathrate hydrate phases. But what we are seeing here, if we can keep the temperature pressure constant also, but if we just change the composition of the ice, that also impart a role here and selectively can give structure 1 or structure 2 Clathrate hydrate. And secondly, here we are saying that structure 1 is giving us, if you can take the structure 1 Clathrate hydrate, it is inhibiting the kinetics of crystalline ice formation. But if we take structure 2 Clathrate hydrate, that's actually facilitating the crystalline ice formation. So by preparing a specific structure of Clathrate hydrate, we can actually control or tune the hexagonal ice formation in ice matrix. So for the future direction, what we can say that as we have explored a few ethers here, we can go for other cyclic or non-cyclic ethers, and also we can go some nitrogen containing molecules like pyridine or other molecules to prepare the Clathrate hydrate and see the effect. Also, like here we are all probing the structures by using rare study, but if we can take the help of some cryo-EM microscopy or read study to understand the structural conversion. And also, like here we have seen that two structure is forming at two compositions, structure 1 and structure 2. But if we can transform the structure from structure 1 to structure 2 or vice versa, that can also be monitored using cryo-EM or read. And lastly, we can study the photochemistry inside the cage to irradiate the molecule by using our this deuterium continuum source, and to see the reaction inside the cage and study the photoproducts. So yeah, so last of all, so I would be thankful to SAT to giving me the opportunity to study these experiments and always encouraging me for this study, and also to all our lab members for their support, and specifically all our ICE lab members who have spent their all time and to build this instrument, maintain this instrument, and so that it still performs so well. And also I'm grateful to our funding agencies, DST, ACRB, IITM, and also the Swarna Jayanti Fellowship, which has helped us to build this instrument and maintain this instrument in this condition. Thank you all. I am open to any kind of question.
Thank you so much, Soham, for very nice and interesting lecture. So now the floor is open for the question, and please ask.
First of all, Soham, you have done a fantastic job, great work, a lot of painstaking effort in creating this data. And of course, much more, many more papers are coming. All that is very nice. And I am so glad that you have, within the group, you have created another group. And I see that, well, I also saw Soham here. And apart from others, I already mentioned Gaurav, Robin, and Robin. So this is a very nice thing. Now, if you start thinking about this work, because several other things are going on in the group, it may be of use to other people to know. Vijesh has done a phenomenal amount of work in making sure that these structures can be seen under cryo-EM, and those here, Rabin and Jobin and company we were happy to know that we have just today I was seeing the crystal structure of Clathrate hydrate determined by electron diffraction. I was seeing Harshita's data. Phenomenal amount of work is happening. So all that is beautiful and is coming to some good shape to understand that, making sure that Clathrate hydrates are indeed forming in vacuum, and that is a fabulous thing. And so obviously hundreds of molecules exist, lots of papers are possible, And so that is a great thing for Vijesh and Soham and Harshita and all the people. But the key question then would be that what are the new insights that you are providing in comparison to what we know in the clathrate hydrate story? Well, you are putting new molecules and making clathrate hydrates, which were not known to form, as you have just now shown. But if you look at this clathrate hydrate area as a whole, what new insights are you getting? I would first look at that question as a very important central question. Now, probably this is a group which is having capability that no other group in the world has. You have spectroscopy or microscopy or scattering, all of these along with computations. So let me address that, maybe to open your mind, also to tell us what have you been thinking about? What's new that you are revealing?
Okay. So, like as you talked about, one thing is this structural conversion and transformation of Clathrate hydrate. That is a unique thing because we are seeing like usually in high pressure condition, people are using some other gas promoter molecule or something to perform this conversion. But here we are doing in a cryogenic temperature condition where the entropy is very, very low. We are seeing this transformation here. And one thing that what we have seen that we can create a structure 1 or structure 2 hydrate and we can transform that. So we can use this molecule to separate a mixture of molecules. So we can take 2 or 3 molecules and then separate that molecule, and we can say that okay, so this molecule is particular at that condition is going to a small cage, another molecule is going to a large cage. So we can separate out those molecules from a mixture. So and that can be in situ monitored by using IR spectroscopy, which we have read. We can use that, and also we can do some trial.
So great. So that's an insight that how these transformations occur. This is systematic. You are in a position to not to see just the structures, but how the structures evolve. Now, does it also tell us, is it possible for us also, I mean, I would like Vijesh's intervention as well, to know at what time scales can you really make it? Meaning I'm talking about, this is now clathrate is a crystal, you see, is a crystal from amorphous to crystal. So if you have the capacity to see the crystal formation, crystalline material formation, what is it? Is it happening at like NaCl crystallization? Is it like H2O crystallization from liquid to water? There's a time scale for all these. Is it possible for us to understand this?
So if we can create the Clathrate hydrate in situ in the cryogenic setup or our STM setup, we can take water and the gas molecule together and then heat it at a particular damping rate, and we can continuously record the spectrum. But I think this kind of nucleation happens in microseconds. So
good. Maybe Vijesh has something to say. Before that, I want Harshita just to flash her structure after this particular meeting, just for everybody's excitement, so that additional thought can happen later. Vijesh, if you have something to say.
Yes, actually can be done, but we need this system like ultra-crystal electron dynamic, which people are doing to see the real-time photochemistry like photodissociation. That only can give us this time scale, because it will just snap the picosecond or femtosecond scale changes that can give me. So this kind of experiment only people are doing in SLAC and Brookhaven, that national laboratories.
So we are talking about time resolved X-ray.
Yes, yes, sir. Yes, sir. Time resolved diffraction.
Yeah, okay. So this is possible today in a microscope. Is it possible to have electron beams pulsed at that time scale?
Yes, that is possible. Like people are doing electron pulse by generating by photons. So what they're using? They're using photon
right.
and they're creating electron pulse. So that
Fantastic.
is.
Thank you. Thank you. I stop here for want of time and let everybody ask questions. Yeah, thank you.
Hi, Soham.
Hi, Bhaiya.
Yeah, nice presentation. So I have two questions. The first question is regarding the photochemical reaction, photochemistry, which you are talking as a future direction. So what kind of different photochemistry this clathrate hydrate could give other than kind of a matrix, ice matrix? That's my question number one. What difference basically it will give, or what you can expect from that? Because they are already in a kind of a cage, that is correct, but it is a specific cage, that's also correct. But what kind of difference it can give?
Okay, thanks for asking me question. So the thing is, in water matrix, we are taking water molecule, another with the another molecule, and then photodieting that molecule to give some photo products. Now, when the molecule is encased in a clathrate cage, then we are isolating a single molecule inside the cage. So, in this kinetics procedure, there only unimolecular reaction can occur inside the cage. Outside the cage, the particular molecule can be reactive with water molecule or any other molecule or the similar kind of molecule and give many kind of products. But when the molecule is isolated inside a cage, it can go only unimolecular reaction or reaction with the hydrogen or oxygen atom in the water cage network. So that is only possibility. So that can give us a significant different product than what we can expect from the water ice matrix.
Okay. And my second question is, we can make a CO2 clathrate. So will that be a good source for this carbon dioxide trap, which we are in absorption platform, which we can think of for reduction of carbon footprints and all? So can we apply in that aspect, or how good it will be?
Yeah, so like already in high pressure regime, people are working with that. So CO2 sequestration or CO2 trapping in hydrate cage. But we can give us their more chemistry or the reasoning, the chemical reason for the process that we can provide, so that that might be more helpful.
Okay, okay, fine. Good. Thank you.
Hi, so nice presentation.
Thank you.
Can you hear me?
Yes, Bhaiya, you are
Yeah.
audible.
Yeah, nice presentation. So I have this query and one suggestion. So query is this: why do you think that this structure one is more stable? Means what is the physical reason? Maybe you can think of.
Okay. So if we look at these structures, so structure one is more tightly constricted. The TMO is getting squeezed, and also there is a host case hydrogen bonding in this case. But in for structure two, there is no host case hydrogen bonding. So maybe that is giving it more stability in this structure and lowering down this binding energy.
Yeah, that's what actually you are missing in the presentation. There is maybe there is a possibility of hydrogen bonding between your guest and host molecule, maybe in case of structure one. That was missing. Yeah. So that could be a possible. You can see in even your IR spectra if you go back, where you are just doing the where you are making crystalline ice in structure 2-1. Here, just see the 0 R spectra in both case. You will see the difference. 0 R spectra at 135. You can see in one case the spectra at 0 R and in another case. There is a slight difference, right?
You're
So
talking
there is
about OH
yes,
spacing?
that one, yeah. So there is a possibility of hydrogen bonding, and that may be a reason like that, making structure one more stable, and that's why it is not breaking. Yeah, that's great. Yeah. Another one is that, okay, like now we're having structure one and structure two. I've seen that a lot of people have shown that CO2 can form mixed hydrate, and no one able to create methane mixed hydrate. Why we not try with the maybe your molecule or maybe other molecule which we are trying in our lab to make a mixed hydrate with methane? I have seen a lot of CO2 hydrate in ultra high vacuum, but not methane hydrate other than our paper. So is it possible to put methane also to form mixed hydrate? With this, it is forming structure one as well as structure two. Is there any possibility? What you see?
That's a good suggestion. We can always try that. And only issue will be the temperature because it forms around after 120, 130 Kelvin. And
Yes.
we can usually form that very low temperature, 30 Kelvin around. So
Yeah.
that we have to handle.
Yeah, it can form at 120, right? Also.
Yeah.
Yeah.
For structure 2, it can form 120.
Yeah. Okay. Great. Yeah. Okay. Nice presentation.
Thank you.
So any other question?
Maybe we should ask, before you conclude anyway, I would like Harshita to show the structure, if that is, that can add some value to everyone, but continue asking questions. We have a few more minutes.
Yes, Soham, I have just one question. Like, what is the volume maximum? What is the biggest molecule we have put in this kind of Clathrate hydrates? What is the volume of that guest molecule so far? Because these are the small molecules.
So guest molecule, maximum size of guest molecule can go up to this 0.6 angstrom, this kind of level. Like if you go for diagonal.
Okay.
Diagonal.
Okay. And what happened? Like, did you try with when you are talking about the composition dependent? So you are taking 1:3 and 1:15. So what happened when you reverse the ratio about the like whether it's forming the clathrates or not? When you take more TMO and less water.
Yeah, we have tried with 5:3 and 2:3 also. So I have not shown that data here. So we have taken the whole range, higher TMO concentration to lower TMO concentration. So what you have seen that at 5:3 and 2:3 also, so TMO is going to structure 1. At 1:3, it is also going structure 1, but below 1:3, as like 1:6 ratio, it is going to structure 2. So 1:3 ratio is the limiting ratio here. Up to that, we can get structure 1. Below that, if we go for further dilution, it will go into the structure.
Okay, so what is the
So,
thermodynamic barrier for like converting structure 1 and structure 2 within this one composition? Like, just thinking like within one composition, what is the thermodynamic barrier which is stopping to convert structure 1 to structure 2 or vice versa?
So, Clathrate structure requires.
Yeah, sorry.
Okay. So Clathrate structure requires more water. So create the water cage and put inside the. Now, it's all about the nucleation process. If nucleation during the nucleation process, the amount of TMO and the amount of water actually determines how the nucleation will go. It will go to the structure 1 cage, or it will make the larger cage based on the availability of the water molecule. Now, we have tried to, as we are getting two structures, we have tried to create the both structure at once, like coexistence of structure 1, structure 2. We have tried also. And
no, is there any chance to convert 1 into 2? Like,
yeah, that's what
by
we
some
have tried,
external
but as
factor,
structure
like
1 is
doing
more.
some.
Yeah.
Is there any?
So we have tried that. Yeah, you please finish.
Yeah, I'm saying like, is there any way or any possibility to convert, like you prepare one structure 1 and convert to the structure 2 by some external stimuli? So what is the energy barrier which is stopping from 1 to going to 2 or vice versa, 2 to converting to 1, like that? Some transition will be there. So is it, what's your thought? Just it come to my mind like that.
Yeah, that's very interesting. Actually, there is a paper on TMO hydrate. They have introduced H2S gas inside this mixture, deposited that H2S gas and shown that structure 1 is being converted to structure 2 in high pressure condition. But what I am telling here is for this structure 1 is more stable than structure 2. So structure 1 is will be less will have less tendency to being converted to structure 2. Another one is to convert structure 2 to structure 1. But structure 2 has more water. It is 1:6 or 1:15 ratio. So it has already has more water. So we can't take out water from the ice mat. We can add more water to make it more diluted and go for the conversion. But as already more water is present there and we can't take it out, so we can't convert structure 2 to structure 1 so easily. So we have tried the vice versa, structure 1 to structure 2, but it didn't come up with.
No, if you were to take the Deepak's question further, if you pull out some molecules, can you convert structure 1 to structure 2? Did you attempt that? Because anyway, it is water rich when you pull out molecules, right? So is that possible is Deepak's question. If I were to...
Yes.
Yeah, we have tried that, the same mechanism, but we didn't get the expected result.
Sure. Okay. So anyway, phase transitions are indeed feasible and it is appropriate conditions. So now, Bijesh, you have a question? Otherwise,
Yes, sir. No, not a question. One comment I have just for Soham. When making this presentation, like why our work is different than others? Because all the people in the world, they are doing on ice or from water gas mixture, this clathrate hydrate formation. We are particularly doing from high density amorphous solid water. So this is what our work is different than others. So please make sure that.
And
In the
I have mentioned this here, Bhaiya.
Mention it like why? Because this is the reason why clathrate hydrate formation is more facile in our system. That's what we are fighting for a long time. So this is the reason we are writing also. So make sure that is.
I have mentioned here. Okay, I will take care of it.
Good. If you have Harshita, can you flash that thing? I saw Soham unmuted briefly, but I don't know whether he has anything to ask.
Yeah, hello sir, how are you?
Hi, as you can see, everything is okay here.
Very nice, sir. Belated happy birthday. I never got a chance to say you in front of you. So yeah, I would say nice work, and it's nice to see that ISAF has evolved a lot and it's changed a lot. So I'll continue my question or maybe query. I would say it's not a question because I don't know much about this field or this section of ice chemistry. Just now it's in the screen, something is flashing. Please go ahead. Whoever is the in charge,
Sure.
I'll continue next.
My screen is visible.
Yeah.
So we tried electron diffraction first with hexagonal ice. And this is the refinement we have done. And this is the first structure which we got that was of hexagonal ice. Then we tried for clathrate hydrate, for which Bijesh Bhaiya gave us the sample. And initially we were getting this type of structure. This was not refined.
By the way, people should know that this is electron diffraction structure. It is done in a cryo-electron microscope. It is in vacuum. All right. Okay.
So initially, this was the initial structure which we got, which was not refined yet. But after this, when we refined it, we were able to see water molecule, carbon of methane, and this disordered THF. So this is the structure which we have right now. Here we were able to fix the water molecule, disordered THF. But in case of methane, hydrogens are still missing because carbon is having
Can
some
you
rotational...
rotate it and show that it is clathrate hydrate?
Yes, sir. Like this, if you can see.
Yeah, beautiful. Yeah.
So this, still we have to fix hydrogens for methane. And this is the structure which we got.
Can you point out what is methane and what is THF?
Carbon for methane. Let me show. Here. And this is disordered THF, which is continuously rotating.
So in the cage of water, this THF is rotating, is what she is saying, at this temperature that we are having.
I think it's having the positional disorder, Harshita.
Yeah, it has
Rotational disorder.
What temperature you are doing this experiment? What kind of cryo temperature?
With nitrogen temperature.
Liquid nitrogen.
Okay, so just to give you a feel that this is happening in the lab now. So it's a lot of excitement for those who are Jobin and all. It will be very happy thing to see that how things have evolved, to see structures.
It's fascinating.
Good. Okay, Somaba, you have something, otherwise we'll close, yeah?
Yeah. So it's first question to Harshita. When you started this depositing the ice film, what was the base pressure generally at the chamber or at the, you are doing it on a grid,
Bhaiya,
right?
this Clathrate hydrate form outside high pressure reactor.
Oh, okay.
So we
Mount,
created it in high
mount
pressure
those
reactor.
crystals on the grid. Sample was prepared by Vijesh Bhaiya.
Okay, at higher pressure, at temperature or?
Yeah, like THF water mixed and 65 bar methane pressure we gave.
Okay, I got it.
Then,
Yeah.
yeah, that's the path.
Okay. All right. Thank you. Soham, so I have a like very general question. In order to create this clathrate hydrate or this pressure, is it important to have oxides? Water is hydrogen monoxide. How it is important to have presence of oxygen in the system?
Oxides,
So water is the oxide.
yeah. So we have tried with D2O also. But
No, it's okay. I mean, D2O, H2, it's the same thing. Is it, can you go away with oxides? Means something else, like hydrates, others, nitrite, or nitrogen thing and others?
So if we were to rephrase that question slightly, Somaba,
Yeah.
instead of H2O, let us say if you take H2S,
Yeah.
can we create a clathrate-like structure?
For example, hydrogen. I'm not talking about ammonia because ammonia is not used.
now I understand your question, Dada. So this what in Clathrate hydrate structure, this water follows this Barnard-Fowler rule. That is a very well-known rule where one molecule can form 4 hydrogen bonds. That rule is
Slowly,
the
slowly.
principle
Say it
to
again,
create
repeat it.
Barnard-Fowler rule.
Okay, okay.
So that is the basic principle to create this hydrogen bonded network in these Clathrate cages to form Clathrate hydrate. Now, as you are talking about this, let's say some sulfide or other nitrogen containing molecule to create such kind of network. So yeah, those Clathrate are present. Like Clathrate structure is a well-known supramolecular structure, and many people are creating. But we are particularly focusing here in Clathrate hydrate. And for making Clathrate hydrate, we need this OH, H2O or OH functionality to create this hydrogen bonding network. There are a category called semi-Clathrate hydrate, where people are using other functionalized group along with OH to create some defect in the structure, or different type of structure where OH hydrogen bonding is present along with NH or something. These are called semi-Clathrate hydrate. Yeah, preparing those will be a tricky one.
Okay. All right. So this question actually it came from because all of you are working in this field. It's very nice. So clathrate hydrate, this kind of any porous structure which has pores, they are beautiful to accommodate. You know, porous chemistry means you can accommodate a molecule into it. So the accommodation also means you can take it out. So you can put it, you can take it out. So in this context, I wanted to know that instead of 300 monolayers or couple of bilayers of ice, if you can embed with something else, which can be easily, changed or change the matrix and to put new molecule in or take it out. So have you thought about, or does anyone work in the world in this direction? That's what I wanted to know through this context, actually.
Yeah, the groups working in high pressure regime, so they are
Okay.
mostly chemical engineering people. So they are mostly about methane and CO2 sequestration in the Clathrate hydrate or gas hydrate structure, commonly called as gas hydrate. So they are mostly trying to particularly trap one molecule and in a particular temperature condition, and then liberate it out at the appropriate condition as their requirement. So there is one procedure to transfer the methane staying in seabed, transferring that methane staying in seabed to the our purpose, useful to use it in our purpose. Those are one kind of study. So yes, we can particularly trap a molecule inside the cage and then just slightly change the temperature pressure condition. We can liberate it out from the cage and use it.
No, so you have done the TPD also, have you?
Particularly for this study, we have not done TPD, but for our previous many papers, we have shown TPD.
Okay, all right. Yeah, I mean, in general, you have done TPD.
Yeah,
but-
yeah.
And
TPD is
all
possible.
these clathrates are made with ice matrix, water ice matrix, or okay.
Yeah, it's all with water.
All right. Okay. Thank you. Yeah. Thank you very much. I'm done.
Thank you. Thank you. I think Deepak, can you conclude? Yeah.
Yeah. Thank you so much, Soham, for very nice and interacting session. So I will again announce the next lecture in few days. So thank you so much again, everyone, for actively participating and making it successful. So see you.
Best wishes. Thank you.
Thank you.